Hi everyone, welcome to our ninth AP Bio Chat. We are getting super close to your exam date. Just a reminder that the first exam date is on May 18th, which gives you, um, you know, a little bit less than 2 weeks. Today, we're going to start with some questions from you guys, and then we're going to go over last week's gene regulation free response question. Just a reminder that if you have any questions, you can feel free to type them in the chat. If you have questions ahead of next week's session, you can feel free to email me. My email address is on this first slide. If you'd like to keep up with the DNA Learning Center and the live virtual labs and bioinformatics sessions that we're holding, you can check out our schedule on our website. You can also keep in touch with us on our social media platforms. So feel free to follow us and reach out to us. So the first thing that we're going to talk about are some questions that I got through email this week. So the first question was with respect to direction words. So one of you asked for the next AP Bio Chat, can you go over the different direction words? For example, propose, justify, describe, explain in the free response questions and how long each response should be towards them. So when you get a free response question on the AP Bio exam, you're going to see these direction words or these verbs, right? And they're going to be sprinkled throughout the question because you know that the College Board likes to turn one question into a gazillion tiny questions. I think that the most important thing to do when you see them, and just FYI, they're going to be in bold and underlined, so you can't miss them, is to calm down and realize that the verbs are actually meant to guide you. They're there to help you. And they help you because they tell you exactly what to do, right? So my first piece of advice is to just do exactly what the definition of that word says to do. And I know that sounds really oversimplified, but we'll talk about examples of these sorts of verbs in a second. And as you sort of allude to in your question, some of these verbs are going to require more words than others, just based on what those words mean in English, right? So for example, if the free response question tells you to propose an experiment, that is obviously going to be a lengthier written endeavor than if they tell you to calculate the mean, right? And so I think part of the answer to your question is innately captured by like what the English definition of the verb is, right? And so I guess there's no definitive answer to your question, but I do want to kind of walk you through some of the most commonly used verbs in this section. So the first thing to sort of recognize is that some of the verbs are going to ask you to produce new ideas, right? And then some of the verbs are going to ask you to just vomit things that you've memorized already, right? So there's not going to be, you're not inventing, for example, how photosynthesis works. You've memorized it, right? And so I think understanding the difference between these two categories is important. So let's talk about that first category first, the part where you kind of have to invent new ideas based on what you know, right? So some verbs are going to ask you to infer new ideas based on your knowledge. And an example of this is the proposal verb, right? So they might ask you to propose a new experiment or to propose a mechanism for some protein or synthesis reaction that you've never seen before, right? Or propose a biological role for some enzyme based on data that they give you, right? So this is an example of a verb where you're going to have to come up with a new idea and defend it using facts, right? So you can't just, in the scientific world, you can't just come up with any argument. You have to support your argument with data. So either data that they give you or facts, right, that you've learned earlier on in the course, right? Some of these verbs will explicitly tell you to defend the inferences they ask you to make. So for example, the word justify crops up on the AP exam pretty frequently, and justify means exactly what it means in the English language to us, right? They are asking you to find evidence, and that evidence is coming from what you learned in AP Bio, right? That will allow you to logically defend the claim that you made earlier on in the question. And then I think this is the easier part of the free response question. So some of the verbs are just going to be very straightforward and ask you simply to regurgitate things that you've learned throughout the course. So they're not going to add that like extra layer of analysis. Instead, they're going to be pretty straightforward and say, define cellular respiration, define active transport, describe the processes by which cells move things in and out of their cytoplasm, right? So these types of verbs are going to ask you more just content specific information and not necessarily have that analysis bit. So generally speaking, students find this first category of verbs more difficult to deal with. So I would just be familiar with this list of task verbs. So I'm not going to read them to you because I'm not going to insult your intelligence, but you can sort of read through this list. I do want to go through the difference between a couple that I think are kind of maybe you would think they're interchangeable, but they're slightly nuancedly different. So I wanted to go through describe versus explain. When you're describing something, you are just I don't want to use the word explain, but you're sort of listing out the key characteristics about the thing that you're describing, right? So describe is usually going to be, going back to your original question, less wordy than an explanation, right? An explanation implies giving information about how something occurs or why a result ends up being the result that you see. So usually explaining requires a little bit of a deeper analysis than simply describing, right? So for example, if I asked you to describe the electron transport chain, you might just say that electrons are moving, uh, you know, in such a way that ATP ends up being produced, right? An explanation would involve, a deeper chemical analysis, talking about things like the electrochemical gradient, talking about the ATP pump, and so on and so forth, right? So in other words, why does the ATP get produced at the outset of the electron transport chain? Not just simply that it does get produced by the electron transport chain. So I think this is probably the trickiest word trap that the AP writers will throw at you. So describe, the sky is blue. It is sunny, right? Explain why the sky is blue is going to be a more complex story. All right, so those are the verbs, and that's a great question, especially considering the fact that your entire exam is going to be based on free response questions. So there is some new information from the College Board that I just wanted to make sure that you're totally clear on, because I think so much of testing is just knowing what to expect and not having any curveballs thrown at you, because curveballs, even, if you know most of the question that they throw at you, a single curveball can sort of throw off your psyche and make you just snowball into failure. So to prevent that, you want to be prepared as much as possible by knowing what to expect. So the College Board has actually posted some online practice exams on their website to simulate test day. So it'll give you access to the new online format. You do need your AP ID number, so that you can get from your teacher, or if your school just has the AP coordinator give it to you, you can get it from the AP coordinator. So what I would highly recommend you doing is logging into the College Board, using your AP ID number and trying out those online practices. And then there is also some new information about the 2 free response questions. So the first one is going to be 25 minutes long. Right. And if you've been practicing with old free response questions, this is analogous to the first free response question on the typical format for the AP exam, where you are given a scenario and an experimental setup with some sort of data, table or graph, right? And then you are going to have to do the following. So this is a pretty straightforward format. You're going to have to describe and explain the concepts that are applied in the experiment. You're going to have to identify and justify the experimental design procedures. So talking about the variables that were used, talking about controls that were used. You have to analyze the data or graph, and sometimes you might even be asked to produce a graph from a raw data table. And so, there's an added layer of knowing which type of graph is appropriate for what type of data set. And then last but not least, you have to use that data to make predictions or make arguments or make claims, right? So they might ask you, based on some set of data, what is happening on a molecular level in the cell, right? The second question is a smaller portion of your exam, but still substantial, right? So it's a little over a third of your exam score. And if you've been practicing with old exams, it is most reflective of the fourth free response question on the old format. So in this fourth question, they're always just it's going to be shorter and more concept based. So you're not having to explain or break down an experiment. So they'll give you a scenario of some sort of biological phenomenon, and they will apply a disruption, right? So in the context of the topics we've been discussing, this disruption might look like a mutation that stops production of an enzyme and what might happen downstream of that, right? So you have to be able to describe the concepts applied. You have to be able to explain the concepts applied. You need to predict what the disruption will do to the biological system or organism as a whole, right? So that's going to hinge on your knowledge of, for example, in the hypothetical I just provided, what does that enzyme do, right? If you disrupt production of that enzyme, then the product that enzyme catalyzes the production of will probably not be produced as much, right? Or at all. And then finally, you have to support or justify whatever argument it is you make with reasoning and evidence. So the reasoning in this particular case would come from your knowledge of AP biology content, right? In the last or in the first free response question, the reasoning or evidence would come from your knowledge and your data analysis. Okay. And then the second sort of question that I have been seeing quite a bit of that I wanted to spend some time on, especially because experimental design is like 2/3 of your exam score. I wanted to really quickly review experimental design basics. So this is what you absolutely need to know. So obviously, when you're designing an experiment, you're seeking to answer some question and expand your knowledge of the universe or the world, right? And so you always start with a question. You always have like a hypothesis. We've talked about null and alternative hypotheses before. Today, I want to focus on the variables and the test groups in your experiment. So the variables are things, vary just means to change, right? It's things that you can change from experiment to experiment. In every single experiment that you conduct, there will always be an independent variable, and this is the manipulated variable. If you have trouble remembering the difference between independent and dependent variable, the way that I remember it is that if I am doing an experiment, the independent variable is the variable that I am changing or I am manipulating, right? And the word independent, serendipitously for us, actually starts with the letter I, right? So the independent variable is the one that I am manipulating. It's the experimental variable, and there can only be one for control reasons. The dependent variable is going to be the response variable, right? In other words, it depends on the independent variable. So we can draw a little arrow from the independent to the dependent variable to show that predicted cause effect sort of relationship, right? And then finally, we need to talk about control variables. So you'll notice here, I'm writing control variables with an S. You can have, and you usually will have, and you should have many control variables in your experiment. There are things that you are keeping the same from experimental group to group, right? So there are things that you use to keep the experiment fair. And I guess, I don't want to mislead you here. You can also have multiple dependent variables. So you can measure more than one effect of changing the independent variable. All right, and then finally, I want to talk to you about the test groups. So the test groups are sorted out based on what your variables are. The experimental group is the group that receives the treatment, right? In other words, it's the one that you are testing the manipulated or independent variable on. The control group is the group that does not receive the experimental treatment. And for AP Bio, you do need to know the difference between a positive control group and a negative control group. I'm going to give you guys an example that I think will clarify this point, but for right now, the positive control means that the effect that you're studying is expected to be present, right? It will be there. The negative control group is the group that you know you're not expecting to see the effect in. In a drug trial, this is what we would call the placebo group, right? So it's the group that gets the phony sugar pill instead of the test medication. Okay, so I'm going to give you guys a quick example. So I'm just going to go ahead and say that you design a new sports drink. It's called Sports Aid, and you want to test its ability to help athletes replenish their electrolytes, right? So the independent variable in this case, what you're testing, is the type of recovery drink provided to the athletes. In this case, the Sports Aid that you're testing, right? The dependent variable would be, in this case, what you're measuring, which is the amount of electrolytes that is in the athletes after some sort of exercise, right? And then things that you would want to control. You wouldn't want one of your athletes to, like, I don't know, do a low impact workout that doesn't really produce a lot of sweat. So maybe one athlete stretches. You don't want the other athlete to like run a marathon, right? Because those are not equal equal exercises. If you do have two people running, right, you wouldn't want to let one runner run 10 miles and one runner run 100 meters, because that wouldn't be fair, right? You would probably also want to make sure that they're running the same pace. You would also want to make sure that the baseline fitness of your athletes was about the same, right? You wouldn't want to have in your experimental group an Olympic athlete and a kid who failed gym class, right? That wouldn't be fair. And then, you obviously would want to control the volume of the drink provided, because if you gave one person, 2 glasses of Sports Aid and only gave the other person a drop of Gatorade, the effects that you're observing might not be because of the recovering ability of the drinks. It might just be because you gave someone more drink than the other, right? Okay. The experimental group in this case would be the athletes receiving your test drink or your Sports Aid, and then your control group. So a positive control is something that you're expecting to see a positive effect in, right? And so a good positive control here would be testing athletes with drinks that are known to help replenish electrolytes already, right? So you could use Gatorade or Powerade or the Pedialyte that your parents probably gave you when you got fevers when you were a kid. I guess some of you are still kids right now, so maybe they still give you Pedialyte. In any case, you would test it against some drink that is known to have this effect of replenishing electrolytes, right? Your negative control would be a substance that is known to not produce electrolytes, right? So electrolytes you can just think of as like salts, right? And we know that water is not going to cause you to produce salt. So a good negative control in this experiment would be water, right? All right, cool. So this is my example. I hope that this was helpful. And then we're going to wrap up the session by going over last week's free response question, which I think brings into play not only gene regulation, but some of the concepts that we just talked about together in the past 18 minutes or so. So this was our question from last week. It was a doozy. So they did this DNA deletion experiment. They built these different genetic constructs, and they looked at the effect on the production of mRNA transcript for this reporter gene that produced something called a prostaglandin, right? And that prostaglandin is a chemical made during tissue inflammation. So conceivably, it helps with your body's inflammatory response when you get like an injury, right? And so the researchers are basically knocking out these different elements or these different genes in this enhancer region and looking for any effect on the amount of RNA transcript for prostaglandin. and therefore conceivably the amount of prostaglandin that they make, right? Because there's a transcription translation connection here. So they're also kind of implying that, right? They're not telling you that this transcript is going to translate into prostaglandin. They're expecting you to know that. Okay, cool. So they ask you to do these things that we've already talked about, right? The verbs that you're expecting. I want to, before we sort of dive into the questions, just look at the data and make inferences about it, right? So the first thing that I want us to recognize is that this top construct right here, let's pull up this pen. Okay, this top construct, ooh, my drawing skills are not great. This is our control, okay? So this is our control group. It is our control group because it is the group that is not receiving the independent variable treatment, right? What is the independent variable in this case? Well, the independent variable in this case is going to be the gene that they're knocking out, right? So each of these is an experimental group, right? So these are our experimental groups. And this is our control group. Our independent variable is the gene that they're knocking out, and then our dependent variable is the amount of mRNA that is transcribed, right? And so when you leave the construct alone, in other words, you just leave it as is, you end up, and the other way that you know that this is the control group, right, is if you look at this graph, this bar right here lines up with the 100%, right? And what does this say on the x-axis here? It says relative level of reporter mRNA percent of control, right? And so if this is 100% of control, then you know that this top bar has to be representative of the control, right? Doesn't that make sense? All right, so now let's look at each of the experimental groups. The first experimental construct, we're knocking out gene 1. What happens? What is the effect when we knock out gene 1 on the amount of mRNA that is produced? Is it greater or less than the control group? This bar is obviously greater than, right? In other words, what we're seeing is that the deletion of this first control element seems to enhance transcription of prostaglandin mRNA, right? And it does so by over 50%, right? So we're out here almost at, I'd say like 175, right? Okay, so we're knocking out 1 and we're increasing transcription of the prostaglandin mRNA transcript. And then conceivably, we're increasing the translation of the prostaglandin chemical, right? And therefore, conceivably, you know, like we're just following this logical train. If prostaglandin is helping our inflammatory response and we're making more of it, perhaps we're doing an over inflammatory response. Does that sort of make sense? All right. Okay. And so our next, so hi Suraj, I see that you are late to our session. It's no problem. You can always just rewind it and catch up with us. Okay, so for the second experimental group, we're knocking out the second gene, right? And then what is the effect on the mRNA transcript? And remember, we always want to compare it to the control group, right? That's what the control group exists for. It's our point of comparison, right? We can sort of see that it's still increased, right? We're still above the 100% mark. We're at almost 150% relative to the control. And so I'm going to just annotate for myself here that knocking out the second element also increases transcription of the reporter gene for prostaglandin, and therefore conceivably will increase the amount of prostaglandin we're making. And if we're increasing the amount of prostaglandin that we're translating, then conceivably we are increasing our inflammatory response, which by the way is not always a good thing, right? So in fact, one of the main problems with coronavirus seems to be that it is overstimulating our inflammatory responses in our lungs and causing damage in that way. So, you might think to yourself, oh, well, isn't more, immune response good? It's not always good, right? In fact, over inflammation it can be deadly. So remember, in biology, a key concept is homeostasis. So even, more is not always better. All right, and then let's look at this last construct. In our last construct, we are knocking out the third element, right? And what happened? Oh, wow, this is a dramatic change, right? We have dropped considerably. We're not even at 50% of our control. In other words, knocking out this third control element decreased transcription of the prostaglandin transcript, and therefore will probably decrease translation of that prostaglandin molecule, right? And if we're decreasing translation of the prostaglandin molecule, conceivably, we are decreasing our body's inflammatory response, even in the midst of injury, right? And what's going to happen if we don't have an inflammatory response? Well, then you might end up with, tissue damage. So again, homeostasis is like a key underlying idea here. It's one of the big ideas for AP Bio. So make sure you keep that in the back of your head. So do you sort of see how breaking down the diagram before you even look at the questions can help orient you just experimentally and get you ready to tackle every single one of these questions? And I'm going to show you just how we do that. So going back, right? They tell you to determine the independent and dependent variable in the experiment. But we already did that, right? We said that the thing that we were changing from construct to construct is which control element we deleted, right? And then what was the effect? What was the dependent variable? Well, the dependent variable was the amount of transcript that we were making, right? And so if we go to our little answer key here, we're changing the enhancer construct, or which gene was deleted, and the response variable is the amount of mRNA that was transcribed. And I've just included a little reminder here that the relationship between independent variable and dependent variable is that we're looking for how the dependent variable responds to the independent variable changing. All right. The construct that serves as the control group we've discussed already, right? So again, by taking a few minutes to analyze the graph before you get started, you can basically now just plow through these questions, right? We already identified that this first construct is the control group. One, because we can sort of see that we're not applying the independent variable to anything. And then two, we can sort of see that if we read, reading is important. If we read the label on the x-axis, we can quickly identify that if this is 100% of the relative level of reporter mRNA as a percent of the control, well, 100% of the control means that this is the control, right? Okay, and then part C says to explain whether any of the possible control elements is an actual control element. So use the data to support your answer. Well, if, we're seeing these major fluctuations away from the control group with elimination of each of these elements, then the data is suggesting yes, right? As we said already, gene 1 deletion resulted in way more mRNA than the control. Gene 2 was still substantially more than the control, and gene 3 was way less than the control. And so we can sort of expand on this, right, and use our words and write a couple of paragraphs here. So, I'm not going to read this to you, but basically what we're saying here is that the data is showing that the transcript production of this prostaglandin transcript is increasing when we are deleting genes 1 or gene 2, right? And then they're showing the opposite effect when we eliminate gene 3, right? I sort of went a little bit above and beyond here. So I basically said from a biological standpoint, this will prevent overinflammation of the tissue for genes 1 and 2. And then for gene 3, because knocking out gene 3 results in a decrease in mRNA transcript, we can say that the gene 3 is enhancing the transcription of the reporter gene. And without it, the transcription goes down a lot, right? Okay. And so, in terms of the biological role for each control element, I kind of explained that in the last answer. So, genes 1 and 2 most likely prevent an excessive inflammatory response. Gene 3 most likely allows a proper inflammatory response to happen. And then combined, they're going to keep each other in check and promote a homeostatic inflammatory response in our cells. All right, that was a lot, but I hope it was helpful. That is the end of our session for today. We are almost at your exam, so please, feel free to reach out via email or via Instagram to let us know if you have any questions that you want to cover in our last session, which will take place next Thursday, May 14th at 2 p.m. Eastern Standard Time. In addition to asking questions, feel free to uh, give any suggestions about what kind of topics you want to cover. At this point, I would just try and get online and do those practice exams. And if once you finish those, I would look at old AP exams, which they post on the College Board's website. And you're going to want to focus on, as I said before, free response question number 1 and free response question number 4. Thank you so much for tuning in today. Please take care, stay healthy and well, and I will see you all next week.